Hello, my name is Abi, and today we're going to be talking about bacterial transformation, which the whole point is basically getting like a foreign object, like a plasmid, inside the cell, like an E. coli cell. So for today's video, we're going to be talking about this type of plasmid, and this type of process is meant to do things like isolating protein, which is what we're doing here with the GFP protein. So first, about the plasmid, it's made up of four main components. The first one is GFP, which is the main protein we want to isolate at the end. Next is ARAC, which is a regulatory protein controlling gene expression in the presence of Arabinose. So what you need to know is that this gene blocks transcription, but with Arabinose in presence, it'll actually change shape and move out, which we'll talk about later. Next is BLA, or BLA, and this is ampicillin resistance. Basically, if there's ampicillin stopping growth of the cell, this will block it and actually allow the cell to grow. Next is ORI, which is basically where replication happens for plasmids and chromosomes. Next are the actual steps of transformation, and the whole point of this is to create adhesion pore zones to allow foreign things like the plasmid to come inside the cell. So basically the first step is adding CaCl2, which neutralizes the negative charge. That basically makes the cell competent, which basically means to make the cell able to accept foreign things like a plasmid. Next, we incubate it on ice to slow down the membrane. Then we heat shock. In this case, we're going to be talking about putting on ice and then moving it to a heat bath to get that temperature gradient. And this increases the permeability of the membrane. Next, we incubate, and this allows for the BLA expression to be shown, which we'll see later on. And this basically is to test if the plasmid moved into the cell by seeing if there's ampicillin growth or no growth. So Arabinose, as I said earlier, this basically gets rid of the ARAC, which blocks transcription. So at first ARAC, it blocks transcription, then the Arabinose binds to this ARAC, which changes its conformation or shape. And then the altered shape of the ARAC makes it so that it leaves the DNA. So now the RNA polymerase can actually perform transcription, which is needed. So lastly, this chart, it just shows the different types of whether you have the plasmid or not, and ampicillin and Arabinose, and if there's growth or glow for each one. So first, we're talking about there's no plasmid in the cell, and there's also ampicillin present. So there will not be growth because of the ampicillin being present without the BLA gene to resist it, because there's no plasmid. And there will not be glow because there's neither the plasmid nor Arabinose. So next, there will be growth for the second one because it doesn't have ampicillin, so there's nothing blocking the growth. And there will not be glow because there's no plasmid or Arabinose. Next, we have with the plasmid and with ampicillin, and there will still be growth because of the BLA gene, which allows ampicillin resistance, so it doesn't matter if there's ampicillin, it'll still grow. However, there's no glow because there's no Arabinose, which makes it so that the ARAC still blocks transcription, so the GFP will not be expressed. Lastly, we have the plasmid, ampicillin, and Arabinose, and like I said earlier, the growth will still occur because there's the BLA gene, which makes it so that there's ampicillin resistance, and there will be glow because we have the Arabinose in the plasmid, which makes it so that the ARAC goes away and there is going to be transcription, which allows the GFP to be expressed. That's it for bacterial transformation. Thank you.